I'm Lara. I'm a PhD student at the University of Sydney, and the work that I'm going to be talking about is some of the stuff that I've been doing over the first year of my candidature, my PhD. I'll give you a little bit of a background first so you can understand what's going on. The background's quite important to the whole context of my project. So first, what's rodent environmental enrichment, which is what was mentioned in the title. It's basically a protocol which aims to enrich the rodent's experiences by making alterations to its home cage environment. So cages are typically, there's a few changes that are made. They're typically larger with a running wheel. They contain more rodents and they have more novelty and complexity in them. And so this is an example of what we do in our own protocol. It's very similar across various protocols, but this is ours, the cage circled. It shows all the different changes. So it's actually quite a remarkable paradigm. So if you go through the literature, you can see that it has beneficial and therapeutic effects on seemingly an array of preclinical models of human neurological disorders. I've listed off quite a few there, but I guess the one of note for my project is its impact on neurodevelopmental disorders, which are of a genetic origin. I guess, but in a lot of these contexts, though, the mechanistic understanding for why environmental enrichment has the effect that it has is often really incomplete. And I guess what my lab, me, hypothesized was that maybe this is due to the immense complexity of some of these models and disorders. And then we went on to question, could we potentially garner some useful mechanistic insights from using a somewhat simpler model of nervous system dysfunction, not necessarily a direct model of a disorder as such? Namely the 10M3 knockout mouse. 10M3 is responsible for early axonal guidance. So as a consequence, when you remove this molecule, you get a very stereotyped impaired retinogeniculate topography. So I'll give you an overview of what the normal visual system is like so you can understand this impairment that we see. So this is a schematic of the early stages of the conscious visual system. So we have the retina here, horizontal section, the DLGN, which is part of the thalamus, which it projects to, and V1, which is one of the earliest higher cortical processing stages. And we have the ipsilateral projections in purple, and they project to this very discrete dorsal medial region here in purple, in the DLGN, which is the bit that I want you to focus on. And then the contralateral projections project to the rest of that. So this is an actual image of a real DLGN coronal section here. We've had the projections labeled with an anterograde tracer injected into the eye. And so we've got the ipsilaterals in purple, like in the schematic, and the contralaterals in green. This is our wild type animal that hasn't had any genetic changes made. But in our 10M3 knockout mouse, we get a very stereotyped change in these ipsilateral projections, whereby they're no longer restricted to this dorsomedial region, but they extend into this ventrolateral region, which would usually only be occupied by the contralateral projections. So that's disturbing the overall topography or representation of the visual world within the DLGN, which is really important for functioning. So curiously though, my lab, this is before me, managed to characterize a corrective influence of early environmental enrichment in these mice on this particular defect. So this is our standard housing. This is the same image that you saw previously of the 10M3 knockout mouse at 6 weeks postnatal. But curiously, with environmental enrichment, we characterized a retraction in these ipsilateral projections in this most ventrolateral area here at 6 weeks postnatal of enrichment. We then went on to look at 3.5 weeks, and we found the same type of effect, but it was smaller in magnitude, which indicated to me when I went back and looked at these results that something really interesting was going on at around about this 3.5 week time point. Some active process must be in place to remove these projections. And we also saw with this retraction an improvement in visually mediated behavior, which is usually impaired in these mice, indicative that this is not just an anatomical change, but it's also functionally relevant for the mouse and their capacity to see the world. So what I first hypothesized was whether microglia, an immune cell of the brain, mediated this corrective influence. And this was based off my knowledge that I knew that there were already mediators of structural neuroplasticity during development and using homeostatic plasticity. And so I thought maybe they're involved in this effect. So this is a microglia. it's had its IBA1 protein labeled in green, which is a cytoskeletal protein, which shows you the overall structure of the cell, and CD68, which is a lysosomal protein, in red. Not so much in this cell. And this is also a microglia, but as you can see, it's got a very distinctive change in its morphology. It's got retracted its processes, become more amoeboid, and it's upregulated the CD68, which is in red. And this is what you see, I guess, when the microglia is responding to certain cues in its surrounding environment. And when it's undergoing phagocytosis, which is one of its functional capacities, it tends to adopt this type of morphology. So, based on this knowledge, I went in and had a look at the microglia in the DLGN of the 10M3 knockout mouse to see what they look like, to maybe give me a hint at what maybe is going on at this 3.5-week time point. So these are some low power images of the DLGN. The top is the standard housing and the bottom row is the enriched housing. What I saw in the standard housing was a regionally specific upregulation of both the IBA1, which is in blue here, and the CD68, which was quite curious because this was standard housing. But then when I went to compare with the enriched housing, this is just a visual observation, you saw that in all the enriched mice, this upregulation was much more pronounced in the enriched mice, which indicated the enrichment was doing something really interesting to this signaling pathway. So these are a zoom in, a higher powered image of these insets, and it allowed me to look at the more nuanced, I guess, characteristics of what was going on. And what I found, the enriched compared to the standard house mice had an increased cell density, a clustering around this region, specific region, and an elevated CD68 expression in individual cells, and a change in morphology. So all of these things are very promising for my hypothesis. I went on and looked at, I guess, a quantitative analysis of this, looking at these phenotypes. I first did, I used a software where I could, I guess, 3D reconstruct my high-powered images, and I counted the cells first in 3D space. And what I found, lo and behold, confirming my qualitative observations, I saw an increase in cell density. Then I went on and looked at CD68 expression by 3D reconstructing the cells using a special software package and then measuring the CD68, which is in purple in these images. And lo and behold, I also found a significant increase in CD68 in the cells in this specific region. And then I also looked at a measure of morphology, which I created, which is called 3D solidity. Basically, how much of the convex hull is taken up by the microglia. This is 3D convex hull. Having a higher 3D solidity indicative of more amoeboid morphology, like what I showed you in the previous slides. And lo and behold, the enriched microglia also had a higher 3D solidity, indicative that they were more amoeboid in general in this specific region. So all of this was very promising, but it didn't give me an actual, I guess, mechanism. It all pointed to the fact that they were activated, something really interesting was going on, but I'm like, okay, I'm going to look directly at phagocytosis. And I was able to do this because I had labeled the neurons with a tracer, which could be taken up by the microglia, and it was resistant to lysosomal degradation. So you could actually measure how much of this tracer was in the lysosomes of these cells to give you an indication of how much they were phagocytosing in this area. So this is an image to show you the tracer in yellow within the lysosomes, which are purple. So the first measurement I took from this, I guess, phagocytosis assay was the density of cells in this region which were positive for this tracer within them. And I found a significant increase in this density of cells with this tracer. And then I also looked at a very similar measure, but somewhat different, which was the proportion of cells in this region which were positive for this tracer. And I found the same thing using a slightly different measure. And then I looked at something somewhat more different, which was basically looking at an overall volumetric measure of how much a phagocytosed volume within the cell, so somewhat different. And I found cells tended to, on average, take up more of this tracer in the enriched condition in this specific region than standard house mouse. It was quite a stark difference, really. So all of this stuff seemed to really support my hypothesis that early enrichment elicits a corrective remodeling of this axonal guidance defect by enhancing the phagocytic uptake of these ectopic projections by microglia. It seemed to support this model. And this is, I guess, an important study because it seems to be the first to show that enrichment has a facilitative effect on an adaptive remodeling actions of microglia. So there's been studies which have shown the impact of enrichment in, I guess, immune context, because microglia are also an immune cell, but there seems to have been no studies to show that enrichment has an effect on their, I guess, more plasticity associated functions. So it's quite novel in that regard. And I guess in the larger scheme of things, in regards to human health, I guess you could question, does this knowledge have downstream implications for understanding enrichment's, I guess, beneficial effects on neurological disorders, especially those characterized by impaired connectivity? So who knows, we'll have to find out. So thank you. Do I have any questions?
Hello everyone, my name is Jerry and I'm one of the technical application scientists for BioLegend in the Australia and New Zealand region. I'd firstly like to thank the ADNF committee for organising this event and giving BioLegend the opportunity to sponsor. I think this is a wonderful initiative and I'm very much looking forward to all the talks today. So BioLegend is a privately owned global biotech company incorporated in 2002. We're headquartered in San Diego, California, and this is a picture of our new headquarters, which opened in mid-2019, along with a picture of our founder and CEO, Jean Lay. Our mission is to enable legendary discovery from research to cure, and we aim to fulfill this mission by developing technologies and manufacturing products across research areas, including but not limited to neuroscience, stem cells, immunology, proteogenomics, and oncology. I'd also like to mention that BioLegend is always interested in forming partnerships with researchers. As an example, we work actively with groups at the Garvan Institutes and also other institutes around the world to test emerging technologies. So if there are any technologies that you'd like to work with us on, or if you would like to discuss anything I talked about today in more detail, please get in touch with me or my colleague Hong Yan, or you can check out our website. I've also listed the contact details of our distributors in Australia and New Zealand, and I will stay around in the chat, so please feel free to message me there. Thank you very much.